Hello, welcome back to Bioclass Bites. In this video, we are still talking about the central dogma of molecular biology, but this time focusing on DNA mutation. Don't forget to subscribe to my channel and like and share this video. Again, in this video, we will be talking about DNA mutation. So we're done with all the steps of the central dogma of molecular biology, and at this point, we are assuming that everything went well, that all the processes from DNA replication to mRNA transcription to tRNA translation to protein-synthesis all went well. However, it is possible that things could not happen the way they're supposed to. So what could happen when things go wrong? So this question has been a major source of inspiration for some of the most well-known fictional characters and stories, such as what happened to Spider-Man, to Ben 10, or I think it's Ben 1000 now, or Ben 10,000, to what happened to the Hulk, to what happened to Fantastic Four. However, all of these are fictional interpretations or fictional answers to this question. In reality, something could really also, something could happen or something could go wrong should our DNA mutate. So do not forget the DNA is the code that from which all those processes are based on or are anchored on. So the manifestation of the proteins in our body are all coded in our DNA. So DNA can undergo mutation, and we define mutation as a permanent change in the DNA sequence of a gene. So mutation in a gene's DNA sequence can alter. Since the DNA is the one that codes for the amino acid, the DNA sequence can alter the amino acid sequence of the protein as encoded by the gene. Now, most mutations are neutral or harmless. These are called silent mutations. It means that they do not have any effect on how the proteins will be manifested and no negative effect on the organism's body. However, when mutations affect the composition of a gene, sometimes they can cause illness or even death. So how do we visualize a DNA mutation? Let's assume that this is the genetic code. Okay? And do not forget that that it is read by tree or triplet code or by codon using three of the nucleotide bases. So if this is read properly, the code makes sense. So the sun was hot, but the old man did not get his hat. So if this sentence represents a gene and each letter corresponds to a nucleotide base, each word corresponds a codon or the triplet code. So if this makes sense, if this is interpreted properly by the cells, then everything will go as planned, will go smoothly. But what if you shift the three-letter reading frame? If you shift how the sentence was read this way. So instead of starting as THE, okay, something went wrong here, so it started with HES. Then the sentence will not make sense. Or if it starts at THE, but it started with TH and then ESU, then this will not make sense. The cell will now produce the wrong type of protein that the gene codes for. So if this is the correct sequence, then the right protein will be made. But if something goes wrong with the reading frame on how the gene is read, then it will produce the wrong type of amino acids, eventually the wrong type of protein. So mutations like that are usually due to mistakes that are made when cells copy their DNA before dividing. So if you still remember, this happens during interphase, specifically S phase of the interphase, and that are not detected by the cell's repair mechanisms. So even if there are some minor mistakes, our cells are capable of correcting those minor mistakes if they are just minor. So sadly, if the mistake is too big and the cell cannot repair it, that's when bad things could happen. So that's one probable cause. Then other mutations may be caused by outside mutagenic agents. So when we say mutagen or mutagenic, they're the ones that these are substances or factors that cause mutation. such as ultraviolet rays. Yes, even our own sun is actually a cause of mutation. Radiation, chemicals, toxins, and drugs. So let's look at different types of gene mutation. So if there is only just one incorrect base, we call it a point mutation. So point, okay, just a point, just a single point. A point or single base modification, okay? This causes a single nucleotide base substitution, insertion, deletion of the genetic material. So if there's only just one mistake, if it's supposed to be thymine and then what was coded was cytosine, then that's a point mutation. Okay, so for example, here we have, this is still a point mutation wherein only one nucleotide base is incorrect. So this is a missense mutation. Instead of coding for adenine, there's a replacement of a single nucleotide. It becomes C, cytosine. Okay, so instead of coding for histidine, this is now coding for proline. So this is an incorrect amino acid which may produce a malfunctioning protein. So this whole protein chain might, amino acid chain which will become a protein might malfunction. So this one is a nonsense mutation. So instead of coding for C, it was T that was coded, which will eventually be read as UAG, which is a stop codon. So instead of having a very long amino acid chain, it will be cut short. So that's a shortened protein. So this one is an insertion mutation. So instead of coding for C, letter A was encoded. Then everything was shifted one reading frame. So instead of CAT, it becomes ACA, then TCA. So instead of coding for histidine, threonine, serine, which is actually an incorrect amino acid sequence which can produce a malfunctioning protein. Then a deletion mutation. So instead of coding for A, okay, letter A was removed, then the entire reading frame was shifted. So instead of CAT, it now becomes ATC, which is actually an incorrect sequence of amino acid. Instead of producing a long chain of histidine, now you have lysine, isoleucine, or isoleucine, which is an incorrect amino acid sequence. Again, can produce a malfunctioning protein. Then you have a frame shift mutation. Okay, instead of starting with C, with this amino acid, with this base, the reading frame started with ATT. Instead of CAT, TCA, ATT, CAC, which is an incorrect. The frame shift of just one DNA base results into an abnormal amino acid sequence. So this is now a wrong amino acid sequence that can produce the wrong protein. Then repeat expansion mutation, okay? So there's this repeated trinucleotide of CAG. So it's just supposed to be CAG, then GTA. Now you have 3-glutamine here, okay? Now, if you're interested on the effect of all those point mutation, I recommend that you visit learn.genetics, specifically this page on single gene disorders. I'll provide the link in the description below. Sometimes the mutation does not only occur in one nucleotide. Sometimes a portion of the chromosome can be read mistakenly or can be coded mistakenly. So these are examples of chromosomal variations based on structure. So deletion, that's a portion of the chromosome, and we all know that chromosomes are coiled DNA, okay, around protein. So there's a huge chunk of DNA that's now deleted. So it's so this for example, this segment letter B is deleted, so that can be bad for the organism's body. Duplication, now there's two B segment of the chromosome. Translocation, there's transfer from one chromosome to another. And then inversion, a portion of the chromosome broke off, turned upside down, and reattached in the wrong chromosome. So the genetic material is now inverted. So instead of A, B, C, D, E, F, now you have this incorrect chromosome um, material. Now, mutations that happen in the body, in body cells, may cause an illness such as cancer formation. And we've talked about this in the previous unit about nature of cancer. So, for example, the breast cancer is actually attributed to a malfunctioning BRCA1 gene. And this gene can actually be inherited. That's why breast cancer can also run in the family. Mutations that happen in the germ cells or egg cells are um, are passed on from parent to offspring. So if the mutation just happened within your body, it can cause cancer. But if the mutation happened within your sperm cells, it can cause chromosomal disorders such as Down syndrome and Angelman syndrome. This, I think you're familiar with Down syndrome. Now, I recommend that you visit the online library, specifically the page on genetic disorders and diseases. So there's a huge compilation there of reports from my previous students about their research on genetic diseases and disorders. Also, you can visit this Noctilucerna 2017-2018 issues. These are research papers of my previous students about genetic diseases. So I recommend you browse those. You can access the link in the PDF in the lecture PDF of this lesson found in the online library. So we have mentioned this earlier that a mutagen is a physical or chemical agent that changes the genetic material of a DNA or the DNA of an organism. So sometimes it's bad because it can increase the frequency of mutations above the natural background level, okay, more than what the cells are capable of repairing. So many mutations can cause cancer, and they can also, so therefore mutagens are also carcinogens. So when you say carcinogen, they cause cancer, okay, sometimes. Okay, so this is the international symbol or pictogram for chemicals that are mutagenic. And this is an illustration on how tobacco smoke can actually cause DNA rupture or can cause problems with the DNA structure. So these are possible effects of radiation to DNA. It can break the hydrogen bond between the bases. It can cause base loss, okay, nitrogenous base loss or nitrogenous base change, cross-linkage, or even single-strand break or protein cross-linkage. So these are all the possible effects of mutagens. So examples of those mutagens are radiation. So even natural sunlight or artificial tanning beds can cause mutagenic changes in your body, can cause genetic mutation. X-rays also. So therefore, we should only have at least 2 X-rays spanned equally across the year. Chemicals such as cigarette smoke or preservatives in processed meats or the burnt part in barbecues can also become mutagens. Benzoyl peroxide, which is common ingredient in acne products, are also mutagenic. Infectious agents such as viruses such as HPV, human papillomavirus, can cause mutation and can actually trigger cervical cancer. And then Helicobacter pylori, a bacteria that can spread through contaminated food, are also considered infectious mutagenic agents. So it's best that we avoid all of this so as not to expose ourselves to mutagens. Now, I've mentioned a while ago that our cells are capable of repairing some minor DNA changes. And this video from TED-Ed entitled What Happens When Your DNA is Damaged will help explain how our cells are capable of doing that. So this is a required video for you to watch, and I'll provide the link in the description below. That ends our video. I hope you learned something new today. Again, don't forget to subscribe to my channel and like and share this video. Till next time, goodbye.